All right, so in this presentation, we're going to talk about a very important tool that we use in biotechnology, which actually comes from a living thing. It is known as a restriction enzyme. So what is a restriction enzyme? Obviously, it is from the macromolecule class proteins, and we know that enzymes catalyze chemical reactions. So they bind to substrates and they speed up their changes, right? Whether they're being broken down or whether they're being built up. Specifically, restriction enzymes, their job is in cutting DNA molecules, and that's basically going to fragment them. And their accurate name is a restriction endonuclease. So when you look at the word endonuclease, ASE tells you it's an enzyme. Nuclea, it tells you that it is material within the nucleus, and endo prefix means within. So literally, it is an enzyme that cuts within nuclear molecules. Naming endonucleases is really interesting, but even more interesting is where do we find them? We do not, as humans, contain endonucleases, but we have found them in a variety of microorganisms. So here, you may have heard of this restriction enzyme. This one is called EcoR1, and that's its full name. The E and the co come from the name of the organism using binomial nomenclature, Escherichia coli, or as you know, E. coli. So it's coming from bacteria. That's where we get the eco from. R tells you what strand it is. So when we look at E. coli species, there have been many different mutations, and so we have to identify which one. And the I is actually a Roman numeral. It stands for a number. And in this case, it's the first restriction enzyme that has been isolated from this species. So here's another example, HIND3. HIN is coming from the Haemophilus influenzae. Then D represents what strain it is, and the three I's represent the fact that it is the third restriction enzyme isolated. You're not responsible for knowing how to name restriction enzymes, and the way we say their names, that's just made up. We're just looking at this and saying, I don't know, HIND3, that sounds about right. But I just wanted to share this information because I usually get a lot of students asking. So what are we going to do? We're going to use these restriction enzymes to cut DNA at specific sequences, and these sequences are very unique in the sense that they are palindromic. So what is a palindrome? To give you some examples of palindromes, mom, race car, Hannah. Basically, a palindrome is a series of letters that is read forwards and backwards the same way. Well, when we're reading DNA sequences, 5 to 3 going left to right, and then on the other side, 5 to 3 going right to left, we can find palindromic sequences. They're read the same way, no matter which direction you're moving. And these sequences are usually about 4 to 6 base pairs long, so they're small. They're very difficult to find. So what does an endonuclease do? So this little enzyme basically is zooming around in the nucleoplasm. Actually, it's not inside the nucleus. It's found mostly in the cytoplasm because obviously prokaryotic cells don't have a nucleus. My apologies. And basically, enzymes will bind to substrates whenever they have the affinity for binding, right? So whenever it finds its matching recognition sequence, then the endonuclease is going to bind and it is going to facilitate the reaction where bond breaking is going to occur on the DNA molecule. Now, this is really important. So here's an example. HIND3 recognizes this sequence. So it recognizes A, G, C, T, T. So if I'm reading the top left to right, now I'm going to read the bottom right to left, and it should read the same way. A, G, C, T, T. Isn't that fantastic? It's palindromic, okay? So when this enzyme comes in, this is super important. Please make sure you make a note of this on your notes. It is only going to make vertical cuts here and then here. Okay, those vertical cuts are happening between adjacent nucleotides on the same strand of DNA. So that means we're breaking those phosphodiester bonds in between the 5' end of one nucleotide and a 3' end of another nucleotide, right? In between here, the restriction enzyme is not doing any work. But when you put a cut here and here, these 4 weak hydrogen bonds, it's not going to take a lot of force for them to break on their own, okay? So restriction enzymes cut between adjacent nucleotides, and then here we're going to see these few hydrogen bonds just break from the pressure. This is a very interesting kind of zigzag cut that is occurring because when HindD3 recognizes this full sequence, it knows to bind and cut between the two A's. Each restriction enzyme is going to have their own recognition sequence, so this is very specific. And there are thousands of different types of restriction enzymes. So you might be asking, why do bacteria have these in the first place? Well, to think about it, bacteria don't have sophisticated immune systems like we do. So when bacteria are infected by something smaller, like a virus, what a virus is going to do is inject its DNA or its RNA. It's going to inject any genetic material. And by having these restriction enzymes free floating in their cytoplasm, it will be able to cut up that viral DNA before the virus is able to, transcribe and translate its viral proteins and really infect the cell. So the reason that these microorganisms have it, they have restriction enzymes as part of their defense mechanism. That's essentially what it is. So how do we use these restriction enzymes in biotech? Well, in order to manipulate DNA, we are going to need to cut it up into pieces. And so one of the things we're going to be doing next week is gel electrophoresis. And so we're going to take samples of DNA, we're going to add restriction enzymes to them, and we're going to cut the DNA up into specific sequences. But all those sequences are now just sitting in a couple drops of a solution. How can we see it? In order to visualize it, we're going to perform that technique, gel electrophoresis, and it is going to allow us to space out those fragments. And so we're going to create this gel, and this gel is going to show us representation which visualizes where these DNA fragments are. And we're going to learn a lot more about this in the future, next week. But basically, we have to have the DNA in fragments in order to study its differences. So if one DNA, if one DNA sample contains 3 cuts in it, okay, that's going to produce 4 fragments, right? So imagine you have a string laying in front of you and you take a scissors to it 3 times. You're going to get 4 pieces from that. So the interesting thing is, are all DNA molecules the same? No, not necessarily. But let's say we're doing crime scene analysis, right? And we have 3 suspects and we obtain their DNA and we cut their DNA with the same restriction enzyme. Now, because they're coming from 3 different people, will we expect them to show the same cut patterns to produce the same size fragments? No, we would not. But let's say we have an unknown sample that we've obtained from the crime scene. We can also run this DNA sample with the same restriction enzyme, and we can compare the cut pattern and see if it matches up or is consistent with any of our suspects. And then that would help us determine with a very strong likeliness whether that unknown DNA sample actually came from that person. So again, more on this in the future, okay? But what you're going to do right now is you're going to do a restriction enzyme worksheet where you're going to practice finding recognition sequences going 5 to 3 on the top and 5 to 3 on the bottom strand, and then practice identifying what kind of cut you're making. So here, this jagged cut that has been produced is called a sticky cut, and there's a reason for that. But sometimes the cut, like for example, let's say this is a different recognition sequence, and let's say the cut is supposed to be between the G and the C. Well, it's going to cut here, right? And then on this side, it's also going to cut here. So the cuts in that case would line up, and we call those blunt ends. Okay, so you can identify whether a restriction enzyme is going to produce a sticky, jagged cut or a blunt end cut. And that's what you're going to do on your worksheet for today. Any questions? Please remember to email me.